Hi everyone, my name is Sua Lee from the Stein Monogastric Nutrition Laboratory at the University of Illinois. I'll be discussing the effects of different watering options on ileal digestibility of amino acids and net energy in growing pigs. This work was presented in 2022 ASAS annual meeting. First off, let's talk about water. What does water mean to pigs? Water is one of the essential nutrients for growth. Water involves in almost all the processes in the body. For example, water excretes metabolic waste, moves nutrients into the body tissues, and aids digest. Water regulates body temperature by helping dissipate heat. Water is also important for satiety and food intake of pigs and for determining the amount of manure. Then how do pigs consume water? Pigs maintain their water levels in the body by consuming their feed or by drinking water. It is possible that, depending on how water for drinking is delivered to pigs, food intake and water consumption are changed, which may affect nutrient digestion and metabolism in the body. Therefore, two experiments were conducted to test the hypothesis that different watering options affect the standardized ileal digestibility (SID) of amino acids, the apparent total tract digestibility (ATTD) of dry matter and gross energy, and concentrations of digestible, metabolizable, and net energy in pigs. In both experiments, diet was formulated based on corn and soybean meal in a meal form. Pigs were on ad-libitum feeding. In both experiments, there were 3 treatments. First treatment was a dry feeder with no water, but a water source was located outside the feeder. Second treatment was a wet-dry feeder with a water nipple inside the feeder. However, there was no drinker outside the feeder. To distinguish the first and second treatments, we will name the second treatment as wet feeder instead of wet-dry feeder. Third treatment has combined system with a wet-dry feeder with a drinking nipple inside the feeder and also a water source outside the feeder. In both experiments, water consumption of pigs was not measured. First experiment was to determine the SID of amino acids by growing pigs. 6 pigs with an initial body weight of 27.5 kg were utilized. Pigs with a T cannula installed in the distal ileum were allotted to one of 3 treatments using a replicated 3x3 Latin square design with 3 treatments and 3 periods in each square to have a total of 6 replicates per treatment. All pigs were fed a nitrogen-free diet during an additional period to determine basal endogenous losses of amino acids. Each period lasted 7 days with ileal digesta being collected on day 6 and 7 for 8 hours using standard procedures, and the ileal digesta were stored at -20. Data were analyzed using mixed procedure of SAS, and the statistical model included water options as fixed effect and period and pig square as random effect. Means were separated using PDF option with 2 case adjustment. Moving on the result, let me set up the slide first. Dry feeder is presented in yellow bars and wet feeder is presented in blue bars. Because the combination of yellow and blue is green, the combined system was presented in green bars. Results from this experiment indicated that the apparent ileal digestibility AID of dry matter in the 3 treatments was about 70 to 71% and was not affected by watering options. The SID of crude protein ranged from about 78 to 80%, and these values were not affected by watering options. Same was observed for the SID of lysine, methionine, and threonine, that different watering options did not affect the SID of lysine, methionine, and threonine. Likewise, the result from this experiment indicated that the SID of all indispensable and dispensable amino acids were not affected by different watering options. It therefore appears that placement of the water source in relation to the feed is not critical in terms of maximizing amino acid digestibility. Moving on the second experiment, that the concentrations of digestible, metabolizable, and net energy in diets were determined in group housed pigs. For this experiment, we utilized 24 pigs with an initial body weight of approximately 50 kg. There were 6 indirect calorimeter chambers, and thus 4 pigs were housed in each chamber. 6 chambers were allotted to a replicated 3x3 Latin square using 3 treatments and 3 periods to have a total of 6 replicates per treatment. Feces and urine samples were collected for 6 days after 5 days of adaptation using the time-to-time procedure. Consumption of oxygen and productions of carbon dioxide and methane were also used to determine the total heat production. Urine samples were collected during the fasting period, and fasting heat production was also determined. Data were analyzed using mixed procedure of ASAS, and the statistical model included watering option as fixed model and period and chamber within square as random effect. Means were separated using PDF option with two-case adjustment. Let's move on to the results. The slide setup is the same as in the experiment 1. Feed intake of pigs was not affected by different watering options. However, the weight of oats was greater if pigs were fed in a dry feeder compared with the combined system. The weight of the feces was not affected by different watering options, and the same was observed for the fecal energy output. The weight of urine was, however, greater from pigs provided water only outside the feeder compared with pigs provided water inside the feeder, indicating that reduced water loss if water was provided in the feeder rather than from a drinking nipple outside the feeder. The urine energy output was less if pigs were provided the feed using the wet feeder compared with combined system. The apparent total tract digestibility (ATTD) of dry matter and gross energy were not affected by different watering options. The total heat production, fasting heat production, and retained energy in diets fed to group house pigs were not affected by different watering options. The concentrations of digestible energy was about 3,600. Concentration of metabolizable energy was about 34 to 3,500. And the concentration of net energy in the diets was about 27 to 2,800. And these values were not affected by different watering options. Based on these results, it was concluded that under the conditions of these experiments, feed intake was not increased by placing the water nipple in the feeder, and therefore digestibility of amino acids, dry matter, and energy was not impacted by water source placement. As a consequence, quantities of digestible, metabolizable, and net energy that pigs obtain from a corn soybean meal diet is not impacted by placement of the water source. However, water loss appeared to be reduced if pigs were provided water only in the feeder. I would like to acknowledge everyone from Dr. Stein's lab. And if you want to learn about the research we are conducting in the Stein Monogastric Nutrition Laboratory, please visit our website at nutrition.nsi.illinois.edu or search Stein and Pig on Google. Thank you for listening.